Hello, good afternoon, ma'am. Thank you so much for joining us. All right, so I think we will start with the next talk and the next part of the Neuro November convention. So good afternoon, everyone. Good evening, good morning, depending upon whichever part of the world you're joining in from. I really hope that you're having a great weekend, getting to learn a lot about different aspects of neuroscience. We have some really amazing and exciting talks and panels lined up for you, which I'm sure you're motivated to attend, especially after watching our previous extremely powerful thought-provoking and insightful talk, which we recently had. So without further ado, let me get straight into it. To introduce myself, my name is Tuti Chakraborty, and I'm one of the co-founders of Stimulus. I'm currently working in the Rehabilitation Institute at Christian Medical College, Vellore, which is in the southern part of India. And I'm going to be the moderator for this session, which is going to be the first invited talk of the Neuro November convention. So if you are in the field of neuroscience, or if you are aspiring to be in it or in the right field, I'm pretty sure that you've heard of our next speaker. I'm talking about none other than the really, truly inspiring Dr. Shubha Tole. So I'm going to just briefly introduce her, and then, ma'am, you can go ahead with your talk. So Dr. Shubha Tole obtained her BSc in Life Sciences and Biotechnology from St. Xavier's College, Mumbai in the year 1987. Followed by which she went on to obtain her MSc and PhD from Caltech, USA. And she also worked in the University of Chicago as a postdoctoral fellow, following which she joined the Tata Institute of Fundamental Research in 1999. Ma'am has bagged many awards, prestigious ones, and she has a long list of them. So one of them, the most eminent one, being the Shanti Swarup Bhatnagar Award, which is India's highest scientific honor awarded by the CSIR, Government of India. She also achieved the Research Award for Innovation in Neurosciences by the Society for Neuroscience in 2008. So with that, ma'am, I would invite you to share your screen and go right into the talk. And to all our attendees, you can drop in a question just like you did for the previous panel in the chat. So thank you so much.
Okay, can people hear me?
Yes, ma'am, we can hear you.
All right, well, thank you so very much for inviting me. I must say I am completely inspired by this whole enterprise project Encephalon. I don't think, when my generation was undergraduate, we had this kind of creativity and to just make it all happen. So kudos to you. I mean, more power to you. You folks are the scientists of the future. So all of the cool things are going to come from you down the road. And I'm very, very happy to share some of the work from my lab with you folks, the work done by my wonderful students. So, people talk of awards as achievements. They're not achievements. They're recognition of the fantastic, exciting discoveries that one was privileged to make with the help of one's team. So, my students and postdocs completely share in every success my lab has had. And it doesn't make you, it doesn't, the work was exciting already. Okay, the fact that somebody later on recognized it is important because society needs this sort of celebration of success and interesting sort of developments in each field, right? Society needs to celebrate that. Okay, somebody's achieved some development in engineering, in art, in literature, in science. So, but what I'm trying to say is that the fun happened before. The fun happened when your student sat at a microscope and looked at some experimental prep and said, there's something really funny going on here. This section has 5 hippocampi instead of 2. Okay, that is the fun. And that is why we sort of, it is for that we live. So let me share some of this journey with you, discoveries over the last few years. And I will share my screen. Okay. I am going to talk about the circuits of sensation. It is the circuitry that brings sensory information of the world to our brains that we use to actually know what's out there. Without this sensory circuitry, we would be, like brains in bowls, unable to interface with the world. It is this circuitry that brings us almost matrix-like sort of, inputs about the world. This circuitry not only gives us all of this and creates this rich world for us to live in, but it also limits us, right? Because the first thing you realize is that if you don't get information in via your circuitry, you don't know it's there. You don't know it's there. I'll give you an example. You know that we see in a narrow bandwidth of the electromagnetic spectrum, a very, very narrow bandwidth. Wavelengths on this side, wavelengths on that side, we don't know they're there. We don't know anything is there if it is emitting beyond the bandwidth that we can see. So, I mean, what is the world like? We think the world is what we receive by way of sensory information. So studying how the circuitry forms, in some sense, is studying the circuitry that gives us our world and also limits it. So with that little philosophical beginning, so this is the Tata Institute of Fundamental Research's Colaba campus, Mumbai, by the Arabian Sea. If you haven't visited, I hope we can host you sometime after this dreadful COVID is past. And this sort of illustrates the senses I'm going to talk about. Okay, you can kind of hear the music. You can, in order to play the piano, you need touch resolution, right? You need your fingers. The keyboard was designed for human-sized fingers. If the keys were very, very narrow, we would not have the resolution to play this note or that note. We would be pressing 2 keys at once. Right? So we create our world to match our sensory resolution. Vision, for example, we would not be able to see the black keys as distinct from each other in the white if they weren't spaced enough for us to be able to see them. So this in some sense represents 3 of the 5 senses. And having this circuitry form in the embryo the way it is supposed to gives us our sensory abilities. Now, sensory circuitry is really our pipeline to the exterior world. This is a image showing all of the nerves in the human brain. Okay, this riot of color and complexity, this is not a model system for anything, but this is what we aspire to study using simpler model systems. All right, I'm going to conduct a little experiment with you, okay? Imagine, if you will, a tennis net. You've all seen one. If you haven't, here it is. Now close your eyes and use your probes, your fingers, okay, to poke at this material, this strange material that you don't know what it is, and answer, does this material have holes? Okay, certainly you can imagine your fingers going through it, right? Your whole arm would go through it. Of course it has holes. Okay, now imagine a mosquito net. And now if you close your eyes and feel, you're going to feel at best a rough surface. You're just going to feel the rough texture, but you're not going to be able to tell with these probes, okay, that this material has holes. You just don't have the resolution. If I give you a toothpick, all of a sudden you made the probe narrow, or all of a sudden you'll be able to tell it has holes, right? So what I'm trying to say is sensory resolution is everything. Sensory resolution is what allows us to sample our world as best we can, as best we can. Now, there are some things that, we're not going to be able to see with toothpicks and so on. And that's why we have creatively invented the electron microscope, the X-ray machine. Why do we use those things? Because they have much, much narrower wavelengths, and those probes can tell us things that the naked eye cannot, or even the assisted regular microscope cannot. Okay. So how does all this arise? How does all of this circuitry get wired up? Now, this is my fanciful cartoon of how a brain can be built. Obviously, this is not how you can build a computer. Yeah, you can buy parts, motherboards and RAM chips and processors and, wire them up. But the brain has to be grown. I mean, would that, you could get a nice bright idea and plant it in the brain when you want it. Yeah, the brain has to be grown from one cell. Okay, so much as the brain fascinated me right from when I was in the 12th standard, the development of it gripped me in some sense even more. The brain functions because of how it develops. Development is what allows the brain to wire up and bring about its function. So development is everything. If the development goes this way or that way, all of a sudden the brain function has changed, right? So here are two little movies that I'll share with you just to sort of, I mean, no matter how many times I look at them, I marvel at this fantastic developmental machine that is an egg. Okay, so here is a cartoon of a human egg, fertilized egg, and here's a cartoon of a fish egg. And let's start. One cell becomes 2, 4, 8, 16, both of them. And then in both of them, you have this ball of cells, which in the human embeds itself in the uterine wall and forms the placenta. Okay, this little blue line here is going to become the embryo. The fish doesn't need to do all of this work with the placenta. And I'm going to stop this here and say, look at these little choppy dots. This is the beginning of what will be the vertebral column. Okay, the elements, this musculature and so on. And the same thing is going to happen in the fish. Watch here. You're soon going to see the beginnings of a nice segmental, bones of the spine, the musculature and so on come on. And let's let the human catch up now. You can see the little limb bud, the little eye. Here too in the fish, you have an eye coming up. The limbs growing longer. Yeah. So this takes 9 months. This, we're at barely 3 days. Okay. So why is one so much faster than the other? Okay. Because the amount of the job they do is different and the end goal is different. Okay? First, the fish comes with the nutrients. The fish egg has nutrients packaged in it, the yolk. The human has to build its placenta and first create the nutrient structure. So in fact, as the embryo grows, so does the placenta. They have to keep pace with each other. And then the human spends a lot of time developing the large brain, the large cerebral cortex that is going to be necessary for the human to function. When the human baby is born, everything is not quite completely wired up in place. A human baby lies there looking awfully cute, but can't even move. Whereas the fish, this is barely 3 days, and suddenly it begins to twitch, muscles already wired up, flick, 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 and suddenly sayonara, right? And it's not just fish, right? Other mammals do it too. A newborn calf will get up on spindly legs and start walking right away. The human doesn't do that because it's born with the rest of its body barely in place all of the time. Large amounts of it has gone into shaping the brain. And these are just sort of different species with different trajectories. But within all of this complex body and brain and organ system matching, the brain has to move its cells around and assemble itself. So this is a slice culture of a mouse brain where some neurons are labeled with this fluorescent red dye. This is from a colleague in the field, an eminent scientist, Kazunori Nakajima from Kobe University in Japan. I share it with his permission. And just watch the cells move. It's just, this happens in our brains as they form. Right? Look at these cells dart around. It seems a little chaotic, but each cell knows where it's going. Each cell knows what it's supposed to do. And each cell is equipped with the sensors and the machinery to execute complex motions so that the neurons of the brain can migrate to where they're supposed to go. And then similarly, the wires, the dendrites and the axons, the input and the output wires grow and connect the way they're supposed to. So this is the magic and mystery that we seek to study. There's a lot of questions or comments popping up in the chat box. I'm not sure. Oh, okay. All right. I'll anytime there is actually a question that you would like me to interrupt the talk for, I'm delighted to. This talk is for you. So it would be okay if maybe the host could interrupt and say, oh, there's a question I need to take, or if the person is allowed to unmute, that's completely fine with me. Okay, so please do interrupt.
Definitely. I will definitely relay the questions to you. Right now, we have just got some amazing, incredible, wow comments coming in.
And you know, no matter how many times I see this movie, I find it amazing and incredible and wow. I mean, this happens in every little mouse that's running around the street, okay? Every insect, every little animal is not just a human thing. Every animal that, you know, develops a functioning system, a functioning body, has a functioning control system. And this has to happen for every single animal to do its thing. Okay? The fascination of this is why I'm in neuroscience. And this is why I'm in developmental neuroscience, because I want to see how it is all created. Okay? So let me show you a few cartoons to, you know, it's wonderful to say, oh, how is the brain built? This was my sort of fascination in 12th standard. This is the extent of the job ahead. Okay, this was a picture that I saw in my very first neuroscience class in graduate school. This is a simplified diagram of the visual system, simplified. This could be a crazy, you know, metro routing system for a complex city, and this is a simplified version. Now, not only that, each neuron, here is a example of a neuron. Each neuron, all of these red dots on these, so all of these are dendrites and one somewhere is an axon. Each neuron connects with other neurons, large numbers of connections. The human brain is thought to have 100 billion neurons, and each of them is thought to make between 1,000 and 10,000 connections with other neurons. So look at all these red dots, right? Each one is where some other neuron is come and connected with this. And then, hello, my cursor. That's why you have all of these wires, okay? So the color code here is simply things going in a direction have one color, and the minute it changes direction, it changes color. So these are all large tracts of axons, large wires that are connecting. This, as I said, is not a model system. You cannot study this, nor will somebody kindly give me their brain to study. So we use the model system of the mouse, in which we can study how sensory circuitry is wired. So here's a section of a mouse brain. This is a section of a brain where the sensory pathway is fluorescent green. And you can see this bundle that courses under this blue strip. The blue strip is the cerebral cortex. And you can see that these green neurons originate from here deep within, and they curve and then they course under the cerebral cortex. How these nerves make it there and how they wire up is the subject of what I'm going to share with you today. All right. Now, sensation has to have a good resolution, right? You all know that when you want to get a better picture, you go out and buy a, more megapixel camera, right? The more the megapixels you devote to something, the better the resolution you're going to have, right? Okay. Similarly, if cortex is the processor, okay, your cerebral cortex is the processor, if your sensory nerves connect to a bigger patch of cortex, you're going to have more cortex processing signal from that area, right? Let's consider the touch space, okay? If sensory nerves from my arm are going to go to the brain, if they spread out over a larger patch of cortex, I'm going to be able to have better resolution in my arm. Resolution means if you close your eyes and somebody touches you with two fingers, are they two fingers or one, right? That turns out that you have excellent resolution in your fingers. You have excellent resolution around your lips, okay, where two very nearby points will be perceived as different. The same distance, somewhere here will not be perceived as different, okay. That's because the brain has limited resources. Cortex is limited in its size, right, finally. So it's a very clever strategy. This is not a nice uniform one-to-one mapping of every bit of your body onto your brain. There is a mapping, but it's not the same density. If I showed you a picture of how the body would look proportional to the area of brain it connects to in terms of sensation, here's what we would look like. Okay, so this is telling you that greatly, overwhelmingly, our cortex processes are mouth, the front of our face, our tongue, certainly our hands, and very, very disproportionately small is the rest of our body. Okay, this makes good sense, right? So similarly for the visual system, the fovea, the very center, the high density, where high density photoreceptors are, that occupies a large part of the cortical territory. So you have high resolution in some parts, low resolution in other parts. And this is an effective strategy for getting the job done effectively. Otherwise, cortex would just be uniformly divided, and in areas of your body where you don't really care very much about resolution, you wouldn't, you would have resolution. And here, where you're really sampling the world, you wouldn't. Okay, now in our model system, the mouse, evolution has conserved this mechanism. Okay, the same mechanism applies, the same strategy applies, and for the mouse, not only are the forelimbs important. You've seen mice sort of hold things with their forelimbs and their upper and lower jaw, but they have whiskers, and the mouse whiskers, in fact, sense the world, much as our fingers sample the world. Okay, Peggy Mason in her talk this morning mentioned how when two mice meet each other, they sort of exchange a little whisker shake. Okay, so the mouse uses whiskers the way we would use our fingers. And indeed then, if you represent the mouse scaled to the area of cortex that its sensory cortex, that its skin surface connects to, here's what you will get. A large upper jaw, lower jaw, basically the same oral mouth parts, larger forelimb representation, not so much hind limb and rest of body. But look at this, what are these? Okay, this is the whisker pad. Whiskers are very precisely connected up with the brain. And there are not a random number of whiskers. Each of these whiskers have actually names and numbers. Okay, it's very interesting. Let me just show this to you. This is a schematic of the pathway from the whiskers to the brain. It's not a direct pathway. It goes through a stage in the hindbrain, a stage in the thalamus, and then the brain. Okay, but basically each whisker accesses a patch in the brain. So the C2 whisker and the C3 whisker, for example, which means that if you stimulate one whisker of the mouse, you're going to primarily be able to record from one part of the cortex. And in the cortex, these structures have been given an interesting name. They've been called barrels, the cortical barrels. Okay, are there really barrels in the brain? Okay, these were named by Woosley and van der Loos. And I think they must have liked beer, because this is what stacks and stacks of beer barrels look like. And if you look at the outer surface of the mouse brain, okay, if you flatten it out and stain it up for cytochrome oxidase, that's an enzyme that's found in mitochondria. And this is particularly dense in cells that are using a lot of energy. Here's what you see. Okay, so these are the barrels. This is the primary barrel field. And you can see the A, B, C, D, E rows and the 1, 2, 3. Right, so you can do, so why am I belaboring the point? Because barrels are easy to identify. And looking at whether, if you cut out two whiskers, then do the barrels go away or not, it becomes a model system where you can do a perturbation and you have a readout. You have a readout of brain wiring, okay, by looking at the barrels. Okay, so what did we find? We found a gene that controls the formation of the sensory pathway that eventually creates the barrels. Okay, the brain does not have this barrel formation until the sensory nerves go in, and they are whisker-specific sensory nerves. So when the sensory nerves enter the cortex, okay, whisker-specific input will cluster in the middle of cortical cells, and the cortical cells will form these circles, okay, or called barrels, right? So until then, the cortical cells are sort of uniform. It's only this input that comes in that, you have whisker-specific. Cool? Are we cool so far? Should I take a look at the chat box?
So, ma'am, we do have a question come in.
Mouse unculus. It's not called a homunculus, okay? Mouse unculus. Okay, yes. Can sensing be enhanced by training? Wonderful. Okay, for this, I'm going to ask you to come to the Tata Lit Live Festival, which is ongoing. Tomorrow, an amazing neuroscientist, David Eagleman, will talk about his book, Live Wired. And I have the privilege of interviewing him, being in conversation with him tomorrow at 11. Just Google Tata Lit Live and you'll see this. Or if you follow me on Twitter at Shubha Tole, I will soon post information about this. Yes, resolution sensing can be enhanced. Not only that, he's pushed the limits of what sensory perception can do and where it can take you. It will boggle your mind. I strongly encourage you to come. Yeah. Okay, so these are controls normal mice to which nothing has been done. And I'm summarizing here the work of a wonderful student, Ashwin Shetty, who is now doing a postdoc at Harvard. Ashwin, there. Ashwin showed that when you knock out this gene, okay, this is the transcription factor LHX2, when you knock out this gene in the cortex, you lose the barrels. Here it is in a flat mount, and here it is in section. Remember I said that the cortical neurons are not sort of circled around the inputs until the inputs come in. Well, here it looks like there aren't any inputs. And here the cortical neurons haven't circled because there haven't been any inputs. Now, the cool thing about this finding, which he published some years ago, it was the centerpiece of his thesis. The cool thing about this finding is that we didn't knock out this gene in the entire mouse body. We didn't knock it out in the nerves that were coming in. We only knocked it out in the cortex. Okay, we did nothing to the nerves. They came from the whiskers, from the hindbrain, from the thalamus, nicely segregated, you know. But when they hit the cortex, what happened? Okay, what happened? I hope knockout technology is something that's familiar to everybody. If it's not, maybe the host can just let me know and I'll describe it a little bit.
It would be great if you could describe it, because we do have a very diverse set of people now. So just in brief.
All right. So if there are people who have no idea what a knockout is, I'll be happy to say it. I'll use a metaphor. Okay. Suppose you folks have never ever seen an alarm clock. Okay. No, wait. CRISPR-Cas9. You know, we were doing gene knockouts long before CRISPR was a thing. Okay. Suppose you've never seen an alarm clock, and I'm betting many of you may not have because you all have cell phones, right? But imagine those old style alarm clocks in Mickey Mouse cartoons. Okay, and imagine one landed in from the sky. And you notice that its hour hand moved and its minute hand moved faster and its second hand moved faster still. And you open it and you look at all the gears. And you go, these gears are moving and those hands are moving. Maybe the gears have something to do with the hands moving. And you say, okay, I'm going to knock out one gear. Okay, and I'm going to see what it does to the mechanism. And you discover when you knock out a particular gear that the hour hand still moves, but the minute hand has stopped. And you say, okay, this suggests, it doesn't show, it suggests that this gear might have something to do with the movement of the minute hand. Because you see, the second hand may also stop. The second hand movement may be linked to the minute hand movement. So your gear may have on another hand, and it's impossible to tell by this very crude experiment of knocking out one gear. Right? So which is why a PhD takes 5 years, because you have to make very precise experiments, very precise hypotheses, ask the question this way and that way, and then in combination answer it. Okay? So broadly, that's the idea of a gene knockout. How it is done is something you can read up now. It can be done by CRISPR-Cas now, but it was done using other technology before. And the important thing is we can knock out a gene in the tissue of our choice, okay, using a tissue-specific driver. So if you knock out this gene only in the cortex and the input nerves are all okay, somehow there's a problem when they enter the cortex. So what is that problem? Okay. This is the subject of sort of Alice in Wonderland like story, and what my student Suranjana Pal worked on for her thesis. What happens to these nerves? So this is a cartoon. Here's where the nerves start. They take this U shape. They enter this gray ribbon is cerebral cortex. They enter the cerebral cortex and then they penetrate it and ideally form barrels. Okay. And here is the section of the mouse. The nerves are coursing under the cortex, and this is an embryo section, so they haven't yet entered the cortex. Okay. They haven't had time to enter. Okay. So now what do we do with this system? Suranjana asked then, how does the circuitry form? And here's what she did in collaboration with a postdoc named Geeta Godbole and a master's student, Tuli Pramanik, okay, who developed a way of labeling these nerves and making them green so that we could see them. Now, in the normal mouse brain, this is a relatively mature mouse, and this is a section, and you can see these clumps, okay, the barrels have formed. Yeah. And I'll show them to you in high mag just because it looks gorgeous. So this down here is a pathway that has just come in, coursing under the cortex. And look at how these thin, like each of them is, an axon or a cluster of axons. And you can see how they've nicely clumped, okay, inside the barrels. Now here is the mutant where our gene of interest is knocked out only in the cortex. As expected in section, there's hide nor hair of a barrel, maybe one little cluster here, but there's no barrel seen. And look here, okay, the pathway has entered fine as we expected it would, but it is somehow unable to penetrate the cortex. And now this made us hypothesize that maybe we have a sort of hostile cortex kind of scenario. Maybe our gene in the cortex is functioning to make the cortex permissive. And when you take out the gene from the cortex, the territory is no longer permissive, the nerves can't enter. Nice hypothesis. If it was hostile then, it would have been hostile back in the embryo when the pathway first came. Okay, this is a sort of postnatal, slightly mature cortex. All right, so they looked in the embryo. And lo and behold, here's what they found. Here's the control. And you can see the pathway is coursing under the cortex, but it hasn't yet penetrated, okay, because it's an embryonic stage. And here in the mutant, contrary to this whole hostile cortex hypothesis, you can see that you have, in fact, a profusion of nerves prematurely entering the cortex when they shouldn't. It's too early, okay? It's too early. The normal pattern is that they course under the cortex and they wait here for a little while the cortex itself assembles, the neurons there are migrating and forming. This waiting period is somehow just short, and there's premature innovation and it's, not right. Why this premature innovation then? Okay, and so it's known in the field that if you mess with this pathway in a manner such that the axons don't find their targets, they eventually withdraw and retract and sort of wither away, which is what you're seeing here. So by just looking earlier in time, our hypothesis has changed now. This is not the primary defect. This is a cause of, very likely to be the cause of, this overgrowth and unable to reach the right target and premature growth and so on. So this is the primary defect. So now we're looking at something else altogether. What would make these axons overgrow? Okay, for that we have to ask what makes them normally stay and not penetrate the cortex early. And it is known that there is a key structure, a key row of neurons that is necessary to make the axons, sensory axons, wait. Okay, this is called the subplate. You can think of it as a park and hold array for incoming nerves. So here is the pathway that I showed you before. And here in high mag is this row of blue cells that form the subplate. And here is where these axons normally come and wait. So Suranjana hypothesized that maybe something is wrong with the subplate. And I'm not going to complete this story because we don't have the full answer, but the short answer is definitely something is wrong. Here is the normal tightly packed subplate. And this is a mature brain. So you can see that you see little barrels, even though they're not stained up, you can see little clusters here. Okay, so this is a normal brain. And this is a tightly packed subplate. And here in the mutant, okay, the cortex specific LHX2 mutant, you can see that the subplate is disorganized. It's not packed, it's got gaps in it. Okay, so not only is it disorganized, but also each cell here is a mutant cell. So it may be that LHX2 is required for as yet unknown properties of the subplate that are necessary for these axons to grow. But we know some of the properties. In collaboration with Upinder Bhalla's lab and his student Dipanvita, Suranjana took these mice to Bangalore, NCBS Bangalore, and they sectioned the mice and they recorded from these subplate neurons. And they found that these mutant subplate neurons are way more inactive than the normal ones. And they are inactive right in the embryo, right when the nerves come in and they touch the subplate. So we know some of what the problem is, and it may be that this inactivity is what's causing the axons to not notice there's a subplate and just profusion, grow up to the cortex as if, they didn't know the subplate was there. So this is the sort of precipice on which we are right now. Can you imagine what an exciting time it must be right now for the people doing the next experiment and the next experiment in the lab? Because we don't know exactly what the problem, we don't know which of these properties is relevant, and we're trying to find out. But Meanwhile, okay, so here's a cartoon of the brain again. And meanwhile, while Suranjana was busy looking at the barrels, this is almost like a TV series where something is happening in one city, there's a lot of drama, and then suddenly the camera cuts and some other drama is happening in some other city. So here is our other city or other country, okay, over in the hippocampus. Here, this yellow structure, the structure that records memories, okay, the LHX2 mutant has other problems, okay. What I'm trying to bring out is that the same gene, the same protein, LHX2, it's like a tool, and it does different things in different parts of the brain. And we've been privileged to basically discover a few of them. So while the LHX2 is necessary for subplate properties so that these axons, sensory axons, may grow in properly and not overgrow too early, and therefore then form barrels properly, so that the mouse may feel. So I've used the sensory modality, but this particular mouse, because its sensory pathway doesn't, wire up properly, it overgrows and then it shrinks back, and then the cortex doesn't get sensory information. So although there's nothing wrong with its eyes or ears or its skin, and there's nothing wrong with the pathway of nerves that goes in, because these nerves cannot access the cortex, this mouse can neither feel sensation nor see nor hear, okay, because of one gene that was removed from the cortex. So look at what a profound effect this can have. And this one gene can control an entire, the mouse's experience of the world. Okay. Interestingly, the olfactory bulb seems to be connected. Okay. So the mouse is probably living in its cage in our animal house based entirely on smell. Okay. So meanwhile, while all of this drama is happening in the sensory cortex, over in the hippocampus, okay, in parallel with Ashwin's study, another pair of very talented students were studying what happens in the recorder of memories, okay? And we stumbled upon a mechanism as to how the hippocampus is built. So I'm now going to share with you a tiny vignette of what's happening in the hippocampus. Okay.
So before that, we have a small question in the audience. Someone wants to know, what is the subplate made up of?
The subplate is neuronal. It is made of neurons. It's the earliest born neurons of the cerebral cortex. And they have electrical properties and they make connections with other neurons and with these, presumably with these incoming sensory nerves, we don't know.
Okay, thank you, ma'am.
Okay, so this is a time axis and these are hippocampal stem cells. Each of these is going to produce hippocampal neurons and hippocampal support cells. And what Lakshmi noticed, Lakshmi Subramanian, is that early in development, hippocampal progenitors have high levels of LHX2, and with time in gestation, the levels of LHX2 drop. And this intrigued her because it correlated beautifully with the fact that early progenitors make neurons and later progenitors produce glia. A progenitor is a stem cell. It will divide and proliferate and make copies of itself, but then it will also produce post-mitotic cells, which early are neurons and later on are support cells, okay? This is actually a common theme across the central nervous system, not just in the hippocampus, but in the entire central nervous system, in the spinal cord, in the retina, of all vertebrates, of the mouse, the fish, the frog, the human. Neurons are made before glia, and they come from the same progenitor cell. So this question is called, when the neuron glia cell fate switch, at what point and how does a progenitor, a stem cell, know it's made enough neurons, it's now going to start making glia? Because this decides how many neurons the hippocampus has, right? And how many support cells the hippocampus has. Well, so two very talented students, Lakshmi Subramanian and Anandita Sarkar, took on this project. And they took on, in some sense, two halves of this project. I'll show you in a little bit. Lakshmi would sing beautiful old film songs as she pipetted, and it was beautiful. When her sections were going well, you could hear her sing. And Anandita here is being held back by a colleague because she had a reputation that whenever there was any cake or chocolate or any food product available in the lab, she would somehow manage to get there first and get the biggest piece. So she's being restrained from doing so in the interest of fairness. Okay, so Lakshmi and Anandita took on two aspects of this problem. And Lakshmi showed very beautifully for her thesis that removing LHX2 from these early progenitors induces them to prematurely form glia. And Anandita showed that augmenting LHX2 in these late progenitors prolongs the production of neurons. So together they showed that LHX2 is both necessary and sufficient to control the neuron glia cell fate switch in the hippocampus. Okay, and this was very cool. So not only have we found a mechanism, a central mechanism that decides how many of each to make, okay, but now we have a model where this number can be tweaked and we can ask, well, is it better to have more neurons? Okay, may or may not be, right? So this problem was taken up by a postdoc in my lab, Archana Iyer. And here's what Archana has, it's a beautiful image that Archana has produced. This is a normal hippocampus. All the blue cells are normal cells. And here is a hippocampus with extra neurons, okay? Each green dot with its arbor, and this is Archana, is an extra neuron that wasn't there, that wasn't supposed to be there. And what Archana is working on currently, okay, this is my last data slide. What Archana is working on currently is asking, do these extra neurons help or harm the function of the hippocampus? So more neurons may not always be good, because especially if you've now got fewer support cells, and maybe these extra neurons, which arose later during the glial phase, maybe they grow and they compete with the original neurons, and maybe they mess up the connectivity, right? We don't know, but now we have a model to find out exactly what it means to have a balance of neurons and glia in the nervous system so that it may do its job. The whole motivation of development is, studying development, is that it creates the fantastic computer, which if it's not done just right, finally the function gets affected. Okay, so here then is my summary slide. I've talked about two completely different portions of the brain, one the sensory cortex and one the hippocampus. And turns out that the very same toolkit, LHX2, a transcription factor, seems to be necessary for different aspects in both tissues. And I'd like to close with sort of saying, you know, I've said before that the joy of science is discovering things with your team, right, with your students and postdocs, and that the high moments are when people find cool things. And it's always nice for an advisor when you find that a student has fallen so in love with her project that she's kind of become one with it, and she lives and breathes with it. And here then is a picture of a neuron, a gorgeous, beautiful neuron, and a picture of a glial cell. And here is Anandita when she was feeling like a glia. And here she is when she was feeling like a neuron. I showed this when she defended her thesis. You notice it was the same day. She switched from glia to neuron in the same day. So I'll close now and I'll take questions. Thank you very much.
Thank you so much, ma'am. That was a truly amazing talk, and it was so awesome to see you make science so much fun, especially like the way you communicated it, and you know, it was really easy to understand. So we do have a couple of questions, and I will relay them to you. So the first question we have was, is it just the area which is responsible, or the number of further sensory resolution, or are the number of neurons also responsible?
Okay, let me just read it myself actually. Which is are the innovating neurons differentiate? Which one are you reading? Sorry.
So ma'am, it was a question asked earlier. So the question asked was, is the area of cortex covered responsible for high resolution for sensation, or does the number of neurons also play a role?
I understand better Bhavika's question. Very nice question. For the purposes of this talk, area of the cortex in some sense represents the number of neurons. So scales with the number of neurons. Cortical density is relatively constant, and if your nerves innervate a larger area, you're effectively innervating more neurons. Yeah? So yes, it is eventually its number of neurons, but area is a catch-all way of saying that. You're right. Okay.
So another question that we had was, are the innervating neurons fully differentiated, or are there intermediate stages in their differentiation?
Very cool. So the neurons that innervate are born first before the cortical neurons. And while they grow, they are immature. But by the time they reach, they have begun to the beginnings of neuronal function. And by the time the working period is over and then they penetrate the cortex, they actually need to be doing synapses and making synapses, and they do some activity matching in order to form the barrels properly. So they need to function in order for the barrels to form. Yeah, they're pretty differentiated. In some sense, differentiation is neuron is said to be differentiated as a neuron when it puts out an axon, because then it's definitely a neuron. But if you mean in terms of function, by the time they reach, yeah, they do start showing synaptic activity.
Great. So another question that we had was, what if we somehow increase the time to penetrate the neurons in the cortex? Will that then increase the resolution?
Increase the time to penetrate. So resolution is increased when eventually your incoming signal is spread over a larger processor, more pixels, right? So time would be relevant if in our mutation the growth was slow. But the growth is not slow. It's in fact premature. It reaches and then it goes all the way in. So that's not the problem that we're seeing. So
all right. So another question that we had is, can we make neurons from the progenitor cells to help with rehabilitation in humans?
Very good question. So the work I showed you was regulating the neuron glia sulfate switch in the embryo. But as you probably are referring to, in the hippocampus, in the dentate gyrus reside adult progenitors that normally make adult dent. I don't know if LHX2 converts those progenitors into glia-making progenitors than neuron-making. And I don't know that making more neurons in the dentate would be adequate for the job at hand, because you want to make neurons where you've lost neurons, not only where the other progenitors are. Everywhere else in the brain, we're short on progenitors. So this mechanism is not going to help us in just a regular adult brain, but it offers us insights on how the process is controlled. So taking, say, skin cells, de-differentiating them in a dish, making them induced pluripotent cells, then controlling how you make neurons out of them, that's where this understanding will help.
All right. So another question was, how were the extra neurons identified? Were they abnormally stained green while the others were stained blue, or was a special stain used to differentiate between them?
Okay, very good question. I sort of skipped over that part. We didn't have to go and stain them afterwards. What we did was the mechanism that we used to induce the LHX2 mutation, okay, was in utero electroporation. The reagent that we used to electroporate itself carried a green fluorescent protein. So wherever the LHX2, whichever cell didn't have LHX2, automatically expressed green fluorescent protein. So that's and the control for that is you can put in the green fluorescent protein without inducing the LHX2 mutation. So you can have a situation where you have green neurons that are normal and green neurons that are extra. And in the extra condition is because you've induced the mutation. And that's basically the construct that you put in to do the experiment. If anything
Thank you, ma'am.
Yeah, you can ask me again.
Sure. So if you want to ask more questions, you can put them in the chat. Another question that we have is, just going to go through it. We have more glia in number than neurons. So what effect does the gene have on this number?
Okay. Now, first I've just put my Twitter handle. So if anybody connects with me on Twitter, that's the easiest way of getting to me. I think the jury is out on whether in the hippocampus, the number of neurons, glia outnumbers the number of neurons by that very much. And I don't really have an answer to your question. We are yet in the very beginning stages of trying to understand whether having extra neurons is a problem at all. So we're literally, I literally gave you sort of fresh data where the precipice of it.
So we have another question which says,
Amartya has just pointed out, sorry, Amartya has just pointed out that a recent study showed that the number of glia and neurons are similar. So yeah, that's sort of what I was referring to. Yeah, please go ahead.
So we have another question which says, is it because of this LHX2 gene that we can remember? Because it is responsible for forming memories, what we smell, that is the sense of smell.
So a gene can't be responsible for forming a memory. Okay, a network is responsible for forming memory, and LHX2 controls uh, the number of neurons in the hippocampal network. So it's 2 steps removed from actually forming the memory. And I was referring to hippocampus, which is not the olfactory cortex. LHX2's role in the olfactory cortex is a different story altogether.
All right, so another question. Wow, we have many, that's great. What could be the possible developmental mechanism of adult-born neurons? Is it the same as early? I lost the question. Sorry.
Yeah, no, so I think I already said what I could about adult neurons. The adult stem cells do express LHX2, and we haven't yet tested whether loss of LHX2 impairs their ability to make neurons. We have not yet tested, so we don't know.
So the person asked, is it the same as early developmental neurons?
Oh, so we don't know because we haven't looked, right? No.
Yeah.
Why do we still study neurodevelopment mainly in mouse models? Can't we switch from mouse models to brain organoids? Okay. Organoid technology is wonderful, but it is not exactly the brain in addition. Okay, it is hard to recapitulate entire pathways. Remember, I showed you how the sensory pathway takes this U-shaped turn and then innervates and so on. People are now trying to make assembloids where they take the sensory nerve source and the cortex and assemble them together and try and get the innervation. To recreate a pathway in an organoid is incredibly difficult. And in an organoid, you make innervation without a subplate. The whole mechanism, why is there a waiting period? There's a waiting period because of the timing of differentiation of the cortex. All of that, if the organoid does not have the same control. If you can't recapitulate the phenomenon itself, you can't study it, right? So that's why not organoids. The other thing is organoids are incredibly difficult and fiddly to make. Okay, you have to, and very expensive to culture them in incubators, and you have to culture them for months and months on end. When evolution has already preserved the mechanism for you. So if you want to ask how axon grows, you can ask this question in a Drosophila, a fruit fly, or in a worm, C. elegans, genetically tractable. So you can ask questions of gene interactions much more easily in genetic models than you can in an organoid. An organoid is not the place to do genetics. An organoid is the place to test out what you have found by doing classical genetics, using then CRISPR-Cas in an organoid. So it's a place to test your hypothesis in a humanoid-like system. But it's not a place to generate genetic interactions.
Thank you, ma'am, for that answer. Another question that we have is, apart from LHX2, is there anything else that might also be responsible for the disappearance of barrels? Maybe another axon guidance cues.
Oh, of course, LHX2 is not the only gene. So there are many things required to make barrels. Amongst them being the NMDA1 receptor, which is required for the matching of of firing between the neurons the nerves that come in and the barrel neurons. So just because I mutated LHX2, the barrels went away. That shows LHX2 is necessary for the barrels. It does not speak to 3 other genes also being necessary. So absolutely, it's a very complex system, and it would be dangerous to base it on one gene. Yeah. This is a cool question from Rashi Ladha. The subplate is made up of neurons. In the LHX2 knockout, the subplate is underdeveloped. In the hippocampus, low levels of LHX2 needs to be. How come one protein is playing different roles in 2 components of the brain? Wonderful. Okay, structural differences in the 2 forms of the transcription factor. Don't have to be. Okay, what has to change is simply the targets that the transcription factor binds to. A transcription factor is a tool. It will open up or repress downstream genes. So in each structure, depending on the neuronal context on the other proteins, on what has gone before, and the state of the chromatin, whether it's open or closed, LHX2 may bind completely different targets. And that's actually something that we're doing in the lab. We've divided up cortex and hippocampus, and we're doing ChIP-seq for LHX2 with both. And we're finding different targets. Absolutely. So that's the answer. And thank you for asking.
Thank you, ma'am. So another question is, does stimulus particular neuron is active for response, or a bundle of neurons work together for the response in a stimulus?
Do you mean with the whisker? So when you
Uh,
stimulate a whisker physically, I mean, this is a sort of more general question. Some neurons can be driven to fire by just one input, and some need to reach some neurons need to reach a threshold before they will fire, and depends on how strongly the earlier neuron drives this neuron, whether it can bring this neuron to threshold or not. So there's no one answer for across all circuits, and depending on which system you look at, especially in invertebrates where the number of neurons themselves is fewer, the answers change depending on which system you look at.
Great. So another question is, can we also say or prove that LHX2 also has influence on neuron regeneration in adult mouse in the hippocampus after injury?
A great question. We can't say it, but we can test it, and we haven't done so yet. So absolutely. Now heard about this, the next generation of students in my lab will be answering these questions. Absolutely. Very good question.
All right, so I think the last question for the talk is, there are obvious differences between mice and human brain. How much can we translate this knowledge in regards to humans?
Very good. You tell me, right? In order to do very precise scientific discovery, you should ask the question that can be answered. Now, if you want to ask how one neuron makes a synaptic connection with another neuron, because evolutionary processes are so highly conserved, you don't have to do this in human neurons. You can do this in a fly neuron. The mechanisms are very similar. So how a sensory pathway connects to the cortex is something that in vertebrates, the pathway is similar. I already showed you the parallels between the whiskers and the fingers, right? So the mechanisms that control the sensory nerves in the mouse are going to be the same as the ones that control the human. The evolution has seen to that. That's why we use model systems. That's why it is so effective to use model systems.
All right, thank you, ma'am. So I think the last two questions, we have really enthusiastic audiences, so thank you so much. So I think the last two questions, I would just read them out to you in continuation with the subplate question. Is it possible that LHX2 is distributed in a certain gradient of concentration, like the ones we studied during Drosophila development?
Absolutely, you're right. LHX2 is in a gradient, and we think that gradient is meaningful. We've published a little bit about it. If you look up Muralidharan et al. 2017. This is Bhavna Muralidharan, a postdoc in my lab, who is now a junior faculty at INSTEM Bangalore. So look up her work. The LHX2 gradient is relevant. We think the dropping off of the LHX2 gradient as you go forward in the brain allows motor neurons to form, the motor circuitry to form in one kind of broad nutshell. So yes, many functions. And wonderful, Arpit Mathur is saying that this is his first experience of a neuroscience lecture series. I'm so glad, it's my privilege. Go forth and discover new things.
Thank you, ma'am. And I think the last question, could we see both the hippocampal and sensory neuron subplate interaction in the same mouse with LHX2 knockout altogether?
Yeah, very much so. So the cortex-specific knockout. So if you knock out LHX2 altogether, you don't even get a cortex. There is an earlier role of LHX2, which I won't get into now. Also, the mouse won't get born because LHX2 is needed for forming erythropoiesis, blood cells. The mouse dies in embryonic stages because the blood doesn't form. So this is a very diverse functions of this transcription factor. But in the cortex-specific LHX2 mutant, yes, you have more glia in the hippocampus, and you have the best up subplate. So for a while, two of my students were literally, taking the brain, dividing it in half. Tum ye dekho, main ye dekhti hu.
So we have the last personal question. It was a really enlightening talk. We have a lot of comments coming into the chat. Someone wants to know what made you choose developmental neuroscience, and what do you find most interesting about it? So if you could just end on that note.
I thought I began on that note.
Yes, that is true.
That the brain was the most fascinating thing for me to study, and how this computer is assembled seemed to be the most important question about it. Rather than how it works, does it grow so that its function is enabled? And therefore, and mistakes in its growth will constrain its functions. So it seemed to me that development, which is not really emphasized in Indian education, most of the students, neuroscience interested students who come to TIFR, they want to look at brain function, behavior, mood, emotion, whatever. Those are all outputs. How is the whole thing done? How is it done? So I'm interested in the basic biology of how it is constructed. And the function is a natural consequence. So that's my perspective.
All right. Thank you so much, ma'am. If I would just like to tell the audience, if there are more questions, you could connect to Dr. Tole on Twitter. She has already provided her handle. And could they also reach out to you by email or any other medium?
Twitter is easier. Email will get buried.
Sure, definitely. So please feel free to reach out to her. And ma'am, I cannot thank you enough for coming and giving your time to us this afternoon. Thank you so much. It was a truly enlightening and inspiring talk, and I learned so much, and I'm sure our audience did as well. So thank you so much, everyone, for joining in. Please stay tuned for the next talk.